Swayam Prabha. Digital India. Educated India.
Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different properties of the enzyme in the course Enzyme Science and Technology. And so far what we have discussed about the historical aspects of the development of the field of enzymology. Subsequent to that, we have also discussed about how you can be able to isolate the gene of your interest so that you can be able to clone that into a suitable vector, and then you can be able to use that for enzyme production. And while you are producing the enzyme, you have different options in terms of the chromatography to purify the enzymes. And once you have the purified enzyme, you can be able to use that, or you can actually be able to test that enzyme to catalyzing the different types of reactions. So if you recall, in the previous lecture, we have discussed about the importance of the enzyme in running the different types of metabolic pathways. So we have discussed about the carbohydrate metabolism, we have discussed about the lipid metabolism, and we have also discussed about the protein metabolisms. And all these metabolic pathways, whether they are belonging to the catabolic pathway or the anabolic pathways, are tightly controlled by the different types of enzymes. So in today's lecture, we are going to discuss about how the enzyme is interacting with the substrate and what are the different techniques you can be able to use to measure the interactions. So if you recall, in the previous module, we have discussed about that the enzyme are catalyzing the different types of reactions. These reactions are either falling into the catabolic reactions or the anabolic reactions. But if an enzyme wants to run the catabolic reactions or the anabolic reaction, it has to process the substrate. So what exactly in both of these pathways, what the enzyme is doing is it is converting a substrate into a product, right, with the help of the enzyme. And in this process, the enzyme is interacting with the substrate and enzyme is releasing the product. Now this enzyme substrate interaction, when it occurs, it actually modifies or it actually changes many things in the enzyme substrates. So the first thing what happens is that when the enzyme is interacting with the substrate, if you write this reaction, you are going to say that it is actually going to form the enzyme substrate complex. Then this enzyme substrate complex is actually going to be get converted into the enzyme product complex. You can have the single strand substrate or you can have multiple substrate, whatever the way it ultimately you're going to have the enzyme substrate complex. This enzyme substrate complex is going to be get converted into the enzyme product complex, and then it is actually going to be get converted into the enzyme plus product, and this product is actually going to be released from the enzyme, and that's how it is actually going to be produced. And this enzyme is actually going to return back to the original state, and that's how it is actually going to be ready for processing another molecule of enzyme. So this means the enzyme, when it interacts with the substrate, it actually goes through the different types of modifications or transitions. So let's see what are the different types of modifications it actually brought, right? So in the enzyme, when it interacts with the substrate, it actually changes the size, right? So because you can imagine that the enzyme is probably will be 40 kD, right? and the substrate could be of maybe like 0.5 kD. So if it is like that, the ultimately it is going to be 45 kDa when the substrate is reacting. So it's actually going to change the size. Not only the size, it actually going to change the hydrodynamic surface area. Okay, so it's actually going to change the size in terms of increasing its diameter. So it's actually going to change the hydrodynamic surface area. Apart from that, when the enzyme or substrate are interacting with each other, it is actually going to change the surface chemistry. So it's actually going to bring the either the positive charge, or it's actually going to mass. So it actually can bring the positive charges into the enzyme, or it actually can bring the negative charges in the enzyme, or it may sometime actually going to neutralize the existing positive charges. Okay. Uh, Apart from that, the enzyme substrate interaction is also going to, because when the enzyme and substrate is going to interact with each other, it is actually going to form the enzyme substrate complex. And this, in this transition state, the electronic configuration of the enzyme is also going to be changed, right? So it's going to have the because when the substrate is binding, it is actually going to have the unstable state. So it's actually going to have the different types of the interactions, right? And so this is all about the how the substrate is reacting and interacting with the enzyme. But the central question is that how and why the enzyme is processing the substrate, and what are the different factors which are governing the interaction of the enzyme with the substrates. So as we said, in the past also, that enzyme is made up of the protein. A minor fraction of the enzyme is also made up of the RNA, which are called as the ribozymes. And we are not dealing with the any type of discussion about this class of enzymes. Protein is a polymer of amino acids, and they are very specific towards the substrate and the product, right? So as we discussed right just now, that the enzyme is recognizing the substrate. Due to the presence of amino acid, it provides the specific environment for catalyzing the reaction with the different types of substrates. And the substrate binds to a small pocket within the enzyme. This pocket is known as the active site. The molecules produced by the reaction is called as the product, right? Enzyme activity can be modulated by the non-substrate molecules such as the allosteric molecules as well as the covalent modifications. And how the enzyme substrate recognition works, right? Because before the enzyme is going to interact with the substrate, it has to recognize the substrate, right? So you can imagine that if this is the substrate and this is the enzyme, it is actually going to react at a very small portion of the enzyme, which is called as the active site. So active site is very, very active in terms of recognizing the substrate. And you can see that the substrate is nothing but the three-dimensional structure, right? So it's actually going to have the some of the positive charges, could have the hydrogen bonding donors, it could have hydrogen bonding acceptors and all that. So you can see that these are the hydrogen bonding donors and acceptors, right? And you can have the positive charges, you can have the van der Waals interactions and so on. So first thing what it actually going to decide whether the enzyme is going to recognize the substrate is the geometric complementarity, which means the 3D structure of the substrate is matching with the 3D structure of the pocket of the active site or not, right? And that is the basis of the lock and key model, right? Although in some cases this is not true, but in majority of the cases the enzymes are actually having a predefined three-dimensional structure, and that is going to be recognized as substrate. So you can imagine that there could be many types of substrate molecules which may be matching with one portion of these three-dimensional structures, and that's how it is actually going to recognize. So a typical three-dimensional structure will actually go and fit into this cavity. But that is not enough. It is actually should have the electronic complementarity also. This means it should have the different types of groups. So for example, the positive is actually going to have the interaction with the negative, and similarly the negative will have an interaction with the positive, right? So that's going to be also true. That also should match, because there could be a possibility that you may have the similar kind of three-dimensional structures, but it may not map the interaction. It may not map the electronic complementarity. So, for example, in this case, you have a hydrogen donor, right? In this case, you have the hydrogen donor, and this is actually the hydrogen acceptor. So, when this substrate will fit into this particular cavity, this hydrogen donor and this hydrogen acceptor are actually going to complementary to each other, and that's how they are actually going to form a very strong bond. Similarly, you have the negative charges, you have the positive charges. So if you have a positive charge, it is actually going to meet with the negative charges, right? And similarly, you can have the hydrophobic interactions. So you can have the hydrophobic patches on the substrate, and that also will match it with the hydrophobic patches. So electronic complementarity is very important because it is actually going to give you the affinity parameters. It's actually going to distinguish even the closely related molecules. For example, if we talk about the hexokinase and glucokinase, hexokinase has a very relaxed specificity, so it actually can recognize the different types of substrate, whereas the glucokinase is only specific for the glucose. So it's only going to have the electronic complementarity only for the glucose, not for the other sugar molecule. Whereas in the case of hexose kinase, it's going to have the electronic complementarity for the main type of sugar molecules. And then we have the stereospecificity. So stereospecificity is also going to, enzyme is going to prefer a particular type of stereoisomer in the substrate. So mostly we are actually going to work with the L type of substrate rather than the D type of substrate, because the L type of molecule or isomeric molecules are mostly being present. So this is what I have given in the next slide, that geometric complementarity, the enzyme is actually binding site has a structural complementarity to the substrate. It needs to bind. Then we have the electronic complementarity. So amino acids that are formed the active site or the enzyme binding site are arranged to specifically interact and attract the substrate molecules. And then we have the stereospecificity. So the binding of the chiral substrate and the catalysis of their reaction is very highly specific due to in large part of the inherent chirality of the L amino acids that comprise the enzymes. Now the big question comes that how the enzyme substrate are interacting and what are different modifications actually brought into the enzyme structures. So you can imagine that if an enzyme is there and it actually interacts with the substrate, it actually brings a lot of changes into the enzyme structure, because when enzyme is interacting with the enzyme structure, it is actually forming the enzyme substrate complex, right? And the enzyme substrate complex is a unstable state, so that's why it is actually going to get breakdown and it is actually going to form the enzyme product, because enzyme substrate complex is a very unstable entity. So that's why the substrate is going to be get converted into products, and this is also unstable. So that's why ultimately the product is going to be released, and the enzyme, it is actually going to be reversed, right? It will actually go back to the enzyme is going to be free, right? That's why it's actually going to be interacting with the substrate molecule. Now the big question comes that when the substrate is interacting with the enzyme, what are the different changes it actually brought into the enzyme structure? So the first change what you see is that it is converting the enzyme into an enzyme substrate complex. This means it is actually going to increase the size of the enzyme. So size of the enzyme, right? Size in terms of the molecular weight, right? Which means it actually going to increase the molecular weight, and it is more relevant when the enzyme is interacting with a proteinaceous substrate. For example, if an enzyme is 40 kDa and it is interacting with another proteinaceous substrate, for example with 10 kDa, then when it is actually going to form the enzyme substrate complex, together it is going to be 50 kDa. Even for the small molecule also, for example, if the enzyme is 40 kDa and it is interacting with the substrate of 0.5 kDa, The change may not be very high, but it still be going to change the enzyme molecular weight. Size in terms of also going to be change in the size of the hydrodynamic surface area. So which means it is actually going to increase the size of the enzyme. Say, so it's going to be a lower size, it can actually be able to change the hydrodynamic surface area. It can make it small or it can be able to make it large. So it can actually make the enzyme a little unfolded. It can actually make the enzyme more compact. So the first modification is that it actually can increase the size of the protein. So it actually can increase the molecular weight. It also can change the hydrodynamic surface area. The third thing is that enzyme is made up of so imagine that this is the enzyme which has the different types of active site, right? Different types of groups, right? And it may have the positive groups, it may have negative groups, and so on. So when the substrate is actually going to bind this site, right? For example, if this is a substrate which is actually going to bind and it has negative charges, it has positive charges, and so on, it is actually going to change the electronic configuration of this enzyme. So it's actually going to mask the it is actually going to mask the functional groups, right, or functional charge group or functional groups on the enzyme. So it's actually going to change the mass of the functional group. This means it's actually going to change the surface chemistry. So the third modification is that it is actually going to change the surface chemistry of the enzyme, and that could be in terms of the it may bring the additional positive charge. It can actually be able to bring the additional negative charges. It can actually bring the hydrophobic groups, right? Because now what you see here is that this is an enzyme. Earlier it was having a positive group, it has having negative groups. Now when the substrate is binding, it is actually going to exhibit all the charge what is being present on the enzyme. So if an enzyme is hydrophilic on one side and the hydrophobic on the other side, you can imagine that earlier the surface of the enzyme is positively charged or negatively charged, which means it is polar in group. But once the enzyme is binding this particular substrate, it's maybe getting converted into a hydrophobic surface. So it actually can have the positive, and so it actually can change the polarity of the molecule, right? It either can bring the positive charges or it can bring the negative charges, or it can actually bring the hydrophobic groups. Apart from that, when the enzyme is interacting with the substrate, it also changes the energy level of the enzyme. So it's actually going to bring a lot of changes in the free energy of the molecule, right? So it's actually going to change the free energy. And all these properties, so the first is it's going to change the size, so molecular weight is going to change the hydrodynamic surface area, so it's going to have the increase in size or decrease in size. The third is it's going to change the surface chemistry, and the fourth, it is actually going to play with the free energy of the molecule. So it's actually going to have the modulation of the free energy as well. And all these properties can be studied with the help of the different types of techniques. For example, if you want to know whether the molecular weight is going up or down, you can be able to do like the electrophoresis, right? You can also, if you are going to know whether the molecular weight is changing or not, you can actually be able to do the electrophoresis. You can also be able to do the gel filtration chromatography, and so on. Similarly, if you want to see whether the hydrodynamic surface area is changing or not, then what you can do is you can use the third technique, and that is called as the gel filtration chromatography, right? So you can actually be able to use the chromatography techniques. Similarly, if you want to see the changes in the surface chemistry, then you can actually be able to choose, you can actually be able to use the ion exchange chromatography, because ion exchange is going to be uh, ion exchange chromatography, or you can be able to use the SPR, or the surface plasma resonance, right? And when you are expecting that there will be a change in the free energy, you can be able to do the many type of technique, but you can use what we are doing discuss in this particular course is the isothermal calorimetry, because you can be able to measure the energy of the complex, you can be able to measure the energy of this free enzyme, and if there is a change, you can be able to say that the enzyme is interacting with the substrate. So let's discuss how we can be able to exploit some of these techniques, and you can be able to use for studying the enzyme and substrate interactions. So enzyme substrate interaction, as I said, enzyme is going to interact with the substrate, and it is actually going to use and form the enzyme substrate complex. And in this process, it's going to have the increase in size. So if you have the increase in molecular weight, you can be able to use the electrophoresis. So electrophoresis is one technique that you can use to see whether there will be an increase in size or not, right? So there will be an increase in size. So size is one parameter, right? And the second is you are going to see whether the gel filtration chromatography is so gel filtration chromatography can be used, right? So gel filtration chromatography can be used for measuring the hydrodynamic surface area, right? So you can actually be able to see how much is the size of that ball, right? And then third is you can actually be able to use the ion exchange chromatography in case you want to expecting, or you can actually be able to use the HIC in case you are expecting that there will be, and you can also use the SPR in case you are measuring change in the surface chemistry of the molecule and the surface is interacting. And the fourth is you can also be able to measure with the help of the ITC, and you can actually be able to measure the free energy of the molecule, and you can be able to calculate what will be the change in delta G, and that's how it's actually going to tell you whether the substrate is interacting with the enzyme or not. So let's start with the first technique, and the first technique is how you can be able to use the chromatography technique. In this particular aspect, you can also be able to use because that when the enzyme and substrate are interacting with this enzyme and forming the enzyme substrate complex, it also going to bring the changes in terms of the spectroscopic parameters, right? So it's actually going to bring the additional variations in terms of the pharmacophores, because that as we discussed, right, and when the enzyme is interacting with the substrate, it's actually going to change the surface chemistry, and that may actually be able to change the electronic configuration of the molecules, and that can be mapped even by the spectroscopy. So that is also going to be one of the technique what we are going to discuss. So the first technique is this, second is this, third is ion exchange chromatography, fourth is ICT, and the fifth is spectroscopy, and the sixth is SPR. So let's start with the chromatography. So in the chromatography, we can have the two chromatography technique because we want to measure the size, and we can also be able to measure the charge chemistry or the surface chemistry, right? So we also can measure the charge. So size we can measure with the help of the gel filtration chromatography, and charge we can actually be able to do with the ion exchange chromatography. So let's start with the charge and how the charge can be used and exploit with the help of the ion exchange chromatography to measure the or to map the enzyme substrate interactions. So for explaining this, I have taken an example of the enzyme which is interacting with DNA. So you can imagine that in this case the enzyme is having the positive charges and the DNA is having the negative charges, and that's how they are interacting with each other. So in this particular, so DNA enzyme interaction, so in this approach, the anion exchange matrix is incubated with the DNA and allow it to bind the DNA tightly. Now, the pure protein is passed through the DNA bound beads, followed by the washing with the buffer to remove the unbound protein. Now the DNA is eluted from the matrix either by adding the high salt or with the denaturating conditions. Now the fractions are tested for the presence of DNA and protein, and the eluted protein is analyzed in SDS page and DNA in the agarose. So this technique what we are talking about is more of a qualitative technique, so that you can be able to say whether this particular enzyme is interacting with the DNA or not. You will not be able to measure, or you will not be able to calculate the affinity parameter, which means I am saying that you will not be able to measure the dissociation constant. So that constant Kd you will not be able to calculate. So what you are going to do is you are going to take the anion exchange matrix into a column, and then you are going to pass through the DNA. So when you pass through the DNA, the bead is actually going to have the DNA molecule which is bound, right? Because the bead is positively charged, so it is actually going to bind the DNA molecule. So these are the DNA molecule, right? And when you are adding the enzyme, what the enzyme is going to do is it is actually going to have no affinity for the beads, because enzyme is also positively charged, right? And the bead is also positively charged. So enzyme will not bind the beads, but enzyme will bind the DNA which is negatively charged. So as a result, the enzyme is going to bound to the to the DNA. And then what you're going to do is you're going to elute with the high salt. So in the high salt presence, the DNA is going to be eluted from the beads, and that's how you are going to have the DNA protein complex which is going to be eluted. And when you are going to analyze this onto a SDS page, it is actually going to an agarose. It is actually going to give you the pattern of the protein, and it's also going to give you the pattern of DNA. So when you have that, what you're going to see is that the pattern of elution of the protein is like this, right? So it is eluting, and then you see this is actually eluting more in the center, right? Similarly, the DNA is also going to be elute in the same pattern, right? So it's going to be elute in the same pattern, and since their pattern is matching, that will say that the protein is binding to the DNA. Now, as a control, you are also going to run the protein onto these positively charged beads. Since the protein is already positively charged, it will not going to bind the column. So this is positively charged protein which you are adding into the anion exchange column. It will not going to bind, and it will come out. So that will be a control experiment to ensure that the protein itself is not binding to the matrix. And that's how you are actually going to get a qualitative test whether this particular enzyme is interacting with the DNA or not. Okay. So once you are done with this, you can be able to use the more sophisticated techniques to measure the other kinds of kinetic parameters, and you can be able to measure the other like affinity parameters. Now let's move on to the gel filtration chromatography, and I hope that you still remember that the what is the principle of gel filtration chromatography, right? So the gel filtration chromatography is basically separating the molecule based on the hydrodynamic surface area, right? Which means it's going to measure how much is the water is associated with the surface. And this actually can be a very robust tool to measure when the enzyme is interacting with the substrate. It actually can bring the two different types of changes. It actually can increase the surface area, or it actually can decrease the surface area. So it actually can increase the surface area, or actually can decrease the surface area, which means when the enzyme is interacting with the substrate, it can actually make the enzyme substrate complex, right? And that may have more surface area. This means the enzyme is going to be get unfolded. Then only it actually can have the larger surface area, or in some cases it's actually going to have the less surface area. And when it's going to have less surface area, the enzyme is going to be get more folded, or I will say it's going to acquire a compact configuration. Now, how we can actually be able to study this? So what we are going to do is we are going to prepare a column, right? So we are going to prepare a gel filtration column, right, with the beads, right? And you are going to analyze your sample, right? So first, what you're going to do is you're going to analyze the enzyme, and while you're analyzing this buffer, what you're adding and what you're using for equilibration can actually have the substrate, right? So so you can actually be able to do this experiment in two ways: one without substrate, and the other is plus substrate. So first you can just analyze the enzyme without substrate, and when you see the chromatogram, the chromatogram will be look like this. So this is the volume, this is the absorbance at 280 nanometer, right? And initially you are going to just do without substrate. So okay, so when you do the without substrate, it's actually going to give you a peak like this. Okay, imagine that if this peak is around 13. Okay, so this is a peak at 13 ml. Okay, now when you changed the substrate, so if it is in the presence of substrate, so when you added the substrate, it actually can go in both the direction. Okay, so it actually can go either like this and can show you this way, or it actually can go in the reverse direction, and it can actually be able to show you a peak in this direction. Okay, so okay, now the red curve, what you see, this is a normal, this is the enzyme, right? This is the enzyme without substrate, and this is the substrate when it is making it more towards this, right? So this is small surface area, so this is actually going to be small surface area, and this is going to be large surface area. So basically when the enzyme will make the get unfolded, it will move in this direction. Okay, and when the enzyme is going to be more compact, it is actually going to be in this direction. So many substrates are behaving the enzyme with a different way. Okay, so it is very unpredictable, it is very difficult to say whether the substrate will shift the enzyme in right direction, or whether it will shift the enzyme in the left in direction. But majority of the substrate, when they form the enzyme substrate complex, they actually make the surface area more less, so it actually makes the structure more compact rather than more unfolded. But there are examples where it actually also unfolds the enzyme, and it becomes more and more, it becomes larger surface area. Apart from this, you also supposed to run the substrate alone, because that also should tell you that whether the substrate is interacting, or where the substrate is eluting. So if the substrate is eluting in a very distant place, next has nothing to do with the enzyme, then you are actually be very confirm about this particular experiments. So if I recall again, that you are actually going to have the first, you are going to run the protein. So this is the protein, this is the protein plus substrate, this is also protein plus substrate. it can go in either direction. If the structure is going to be compact, it will go in this direction. If the structure will be less compact, or I will say will acquire the larger surface area, then it will actually go in this direction. And by this you can be able to calculate the surface area, because the distribution coefficient has the relationship of the between the elution volume and the void volume. So the molecular size of a protein is related to the shape of the molecule, and the relationship between the molecular weight and the radius of gyration is that Rg is m to the power a, where a is a constant and depends on the shape of the molecule. So a is 1, which is for rod, a is 0.5 for the coil, and a is 0.33 for the spherical molecules. So when you are trying to do this experiment, you have to do a parallel experiment to determine the molecular size, right? So this is just the qualitative test, right? You can also be able to very precisely be able to calculate the molecular sizes. So you can actually be, what you're going to do is you are actually going to run the different proteins of the different sizes. So you can actually have the larger protein and the smaller protein, and all the 6 protein you can run, and then you are going to have the KD versus log molecular weight as the calibration curve. So you can actually be able to run the calibration curve, and that calibration curve you can be able to use to calculate the molecular weight of the enzyme substrate complex, right? So in this case, the enzyme, if you when you do the substrate interactions, its enzyme is actually going to move either in the left hand direction or the right hand directions. So this is all about the gel filtration chromatography, how you can be able to use the gel filtration chromatography for calculating the uh, for monitoring the enzyme substrate interactions. And what we have said is that the enzyme, since the gel filtration is working on the surface area of the molecule, you can be able to have the two possibilities. When you have the enzyme is actually going to run like this. When you have the so this is the enzyme, and when you have the substrate, it actually can move in this direction, or the substrate can actually be able to move in this direction. So this is the enzyme plus substrate complexes. So either of these conditions, you can be able to have the modification. You can have the mapping whether this is actually being done with the substrate. Because if this peak is true, and suppose that you have taken the substrate as 1 millimolar, right? What you can do is you can increase the substrate concentration from 1 millimolar to 2 millimolar, and then 5 millimolar, and so on. So when you do that, it is actually going to keep shifting in this direction. Ultimately, it is actually going to get saturated at one point, and that's how it is actually going to give you the clear idea that the enzyme is, this shifting is because of the enzyme substrate interactions. So this is all about the how you can be able to use the chromatography to measure the enzyme substrate interactions. In our subsequent lecture, we are going to discuss more about the how you can be able to use the other techniques like the spectroscopy techniques, or the ITC or SPR, and as well as the electrophoresis to measure or to monitor the enzyme substrate interactions. Thank you.